We'll move on to our second talk, which is Ron Milo, and he's going to be telling us about counting SARS-CoV-2 virions in an infected person. So we have a theme of viral quantification today, which is something we haven't heard a lot about in our previous talks. So I very much appreciate the willingness of our speakers to come and tell us about it. Take it away.
Okay.
Please.
Hi, everyone. Great to be here and to follow up on John's presentation, which indeed I think ties together. I'll be sharing my presentation in a second. I'll also say that everything I'll be presenting, I'm happy to be in this forum because I'm stepping beyond my comfort zone in some of the subjects. And I think in that respect, we'll be love to get feedback. And I've added in the chat the link to our paper draft. And so if anybody finds this intriguing, you're welcome to have a look. And I gave open edit permission, so you can put comments, give suggestions, whatever you think could make the science better. So I'll be talking about the total number and mass of SARS-CoV-2 virions, and this is work together with members of my lab, some of them joining here in the session, and with the group of Rob Phillips at Caltech, whom I've been working on for a while. And some of the things we've been interested in, so this might sound like a bit crazy to try and give values for, say, the total number of SARS-CoV-2 around the world. I'll only say that we've been doing such crazy things in the past, and I think it's an interesting thing to try and engage in. You can see some examples. I wouldn't get deep into them, but if anybody's interested, early on in the pandemic, we were trying to collect the available numbers in the literature on COVID-19. So that's something you can have a look at. And recently, we've been looking at cellular turnover in the human body, things about the increase in human-made mass around the world, trying to make a census of biomass distribution on Earth, revisit the numbers of human and bacterial cells, and all of that together from a quantitative perspective is part of a book that Rob Phillips and I wrote about cell biology by the numbers. All of that is just to give a sense of where I'm coming from. So, I'm interested in viruses, but my expertise lies in something else. And in that respect, I think you could probably help me a lot in this presentation. And so, given the 15 minutes that I have now remaining somewhat less, let me just tell you first what are the questions that we're trying to answer. And even if I won't cover all of them very deeply, at least you know the questions, and I'll also give you the key answers that we arrived at. So one is, how many SARS-CoV-2 are in an affected person? Next is, what fraction of the host cells, or as you'll see, we're mostly focusing on lung cells as the interesting one, are infected? What is the stoichiometry between neutralizing antibodies and the spike proteins? What is the total mass of virions in a person that's infected? And in total, in all of humanity, how many mutations are required during an infection? Say from the moment of infection until you're infecting another person? And how often does each possible base pair mutation arise and propagate? Say, for example, if you're interested in, say, this within a variant of concern of the mutation in specific mutation in 484, how often do you expect such a thing to arise in the population? And I'll also say that if you want, you can write comments and throughout in the chat. And if I won't be able to talk and answer, I think Inon Baron, who's from my team, might be able to answer in real time. So because I only have 15 minutes, let me give you the answers already now, even though it's a bit of the fun of deriving, but at least I won't keep you without the answer. So we found about 10 to the 9 to 10 to the 11 RNA copies around peak infection. That's the number of virions, if you like. And that translates into about 10 to the 5 to 10 to the 7 infectious units. And this conversion is also something I'll be mentioning in a second. If we're talking about the whole cells that are infected, even during peak infection, we find that the vast majority of potential host cells are not infected. So only about 1 in a million is infected. And I'll show you how we arrived at that. The stoichiometry between neutralizing antibodies and spike proteins is such that there's an overabundance of about 10 to the 3 to 10 to the 5 neutralizing antibodies per spike protein. The total mass of virions in a person is about 0.1 milligram maximum, 1 to 100. And within all of humanity, if you like, there's about 1 kilogram that's causing civilization to be, at a very challenging time. So that's overall at a snapshot in time. You can also ask about the cumulative mass of virions throughout the pandemic, and we've been calculating that in the link that I've shared with you in the chat. About mutations, we find there's about half a mutation per infection. That's from the moment of infection to the next infection. There'll be a change in about in every 2 such generations, there'll be about 1 mutation. That translates into 3 mutations per month, or 10 to the minus 3 mutations per base per year, which is in line with values known in the literature. And if you look at a specific mutation, such as the ones arising in all sorts of variants of concern, we find they appear every about 200,000 infections that transmitted from a person after it arises novel in that person to another person, which means that given the rate of cases globally, we have that several times appearing every day, every possible base mutation. So that shows you about the spanning of different mutations. Okay, that might have been a bit a lot to comprehend, but let me try now go a bit more deeply into things. First of all, when we're talking about the number of things here, we have to differentiate between, say, genome copies, which are usually measured, through CT or the, real-time PCR, and we're making the assumptions that this translates into what you can call a virion, meaning 1 genome copy would be 1 virion. Although this is something that we think would be very interesting to try and quantify better, because there could be also some naked RNA, like what is the fraction of naked RNA, I think is an interesting open question. And then at the other extreme, there's the infectious units, which are measured by units such as the TCID50, kind of like analogous to the flag forming unit. Those who are coming from virology know it better. And this really relates into what successful virions would be infecting cells in future infection cycles. And these 2 values are quite different. There's a ratio of about 10 to the 4 we find from different sources, and therefore I'll be reporting both. And so if we're talking about how many virions are in a host, you can look at the different tissues where you tend to find them, in the trachea, in tonsils, in lymph nodes, in the lungs, in the digestive system. In each one, you could look at what is known. This is mostly from rhesus monkeys, because there aren't good data at that resolution in humans. And it's an interesting question how similar or different these would be. And you can find for each one what is the range. In terms of order of magnitude, obviously there's a lot of variation, but you can see here about 100-fold variation that has been reported. In terms of RNA copies per gram or infectious units per gram, you can multiply that, but the tissue mass, again, order of magnitude of the human tissue mass of that tissue, and then you get an estimated number. And you can see that lungs are both dominant in terms of having about an order of magnitude or more concentration of infectious particles, and in terms of the tissue mass. And when you multiply that, they become by far 3 orders of magnitude or 2 orders of magnitude at least dominant, and therefore most of the sheer number is residing in the lungs. And we concluded there's about 1 to 100 billion RNA copies per person, and around 0.1 million to 10 million infectious units per person. Only a small fraction, I want to say, of the potential host cells are infected. In order to derive that, you can go through all the different stages here. I think I'll be running out of time, so I won't go very deeply into this, but this would be publicly available and also is available, as I said, on the version that I put on the cloud. But basically, you go through all the relevant multiplications. There's nothing really complicated here.
I was going to say you're actually ahead of schedule, so if you want to take an extra minute or two, it's okay. please.
Okay, I have a lot more to cover. So how many minutes do I have left?
I guess, okay, if you have a lot more to cover, I guess you're 10 minutes in, so you still have, if you,
Okay.
10 minutes doesn't seem like a lot of time, and I'm taking up your time, so I should let you talk.
Okay, it's hard in real time and and I want to show you more interesting things, I think, still. So I'll only say there's like, you can do step by step the calculation. You can hopefully, if you didn't take a share screen, like a snapshot of the screen, you could just look at what I've shared with you, and you'll find all the information. But but from this derivation, we find that there's way more lung cells than the cells getting infected, and therefore we get to this situation. Similarly, you can do the analysis on the number of viral particles and how they're outnumbered by the neutralizing antibodies. So for that, you take the concentration of antibodies in serum and what fraction of them are actually neutralizing versus SARS-CoV-2, and you get about 10 to the 12 neutralizing antibodies per ml, and that in comparison to the number of spike proteins per ml in the relevant tissues, and you find again several orders of magnitude mismatch or larger number. But you can also rationalize or motivate why this has to be the case in order to be effective based on the affinity of the binding, and that is again in this manuscript that I've shared with you. This thing about the overall mass that I've mentioned of about 1 kilogram, that's kind of straightforward. If you take the number of viral particles, you look at peak infection at this value of about 1 billion to 100 billion, and you multiply by that by the number of people that are infected, which is on the order of 1 million, and you reach this 1 kilogram value, which I guess is kind of like a fun fact. I'm not saying it has any, deep meaning, except for using these things, we can derive all the other results that I'm mentioning, that some of them I think could be of more interest. If you look at the overall mass throughout the pandemic, you need to look at how many people are infected in general. And I guess an optimistic scenario would be that we'll be ending with 500 million, and pessimistic one is about 5 billion. And you do those multiplications and you reach that overall will be dealing with about somewhere between 1 ton and 100 tons of the viruses throughout. Okay, and this is why I was running fast, because I wanted to get to this part, which is kind of like my final part about the mutations, the evolutionary implications. So first of all, if you look at the cycles of replication, you need a few cycles of replication from the beginning, where you have just a few virions, to the state in the end that you have somewhere between 1 million to 100 million infectious units. In order to achieve that, you have about 3 to 7 replication cycles. In each one, you have an amplification of 10 to 100 infectious units per cycle. And if you do that multiplication together with the mutation rate per nucleotide per cycle and the length of the genome, you get about half a mutation per lineage per infection. I would mention that this mutation rate has been measured for other beta coronaviruses and not directly for SARS-CoV-2, which is one of the gaps in the knowledge. I'll mention some more in a second. But now here's an interesting thing. If you take that and you multiply that by the number of overall infectious units produced in a person, you find that within a host, you have somewhere between 10 to the 5 to 10 to the 8 mutations per host that are being produced. And if you compare that to the possible mutations, what are the possible mutations? We know the length of the genome, we know the 3 alternative bases in each one, meaning that you have about 10 to the 5, 100,000 single bases that could, mutations that could happen. So you could say that basically in each person, through an infection, we're basically covering the whole space of potential single base pairs. And we're covering some fraction of the 2 base combinations. That's within a host. There's a huge bottleneck when you're infecting the next person, or when you're doing the infection. And from that, you can ask about what's happening through these bottlenecks as well. So as I've showed before, it's about half a mutation per infection, and there's the generation interval, the time between infections, or a few days. And from that, you get what I was mentioning before, 3 mutations per month, roughly. And if you take that over the whole population of infections throughout the world, you're getting, again, the span of variation. I won't get into all that in a second. I would mention that, as I was mentioning, if you multiply that by the number of worldwide infections per day, you're covering all single base mutations, those 100,000 appearing every day. So I think it gives us some sense of the fact that it's very hard to ensure that we won't get variants that are single base differences from existing ones. Okay, let me mention the knowledge gaps, because that's my ending slide, where I want to see if anybody has insight into that. Please reach out in one form or another. This 10 to the 4 RNA copies per infectious unit, we're really interested to know whether it's correct. We could tell you how we derived it, but we're interested to know how does it arise. So any insights on that would be interesting. And similarly about the ratio between RNA copies and viral particles. The yield of virions per infected cell, kind of like the burst size, if you like, for lytic bacteriophages. We're interested to know, again, that this is based on values that were measured for other bacterial beta coronaviruses. If you know of values for SARS-CoV-2, that would be fantastic. The mutation rate, if that has been measured or analyzed by any of you, that would be interesting. And finally, the clearance rate of virions within SARS-CoV-2. We've been using information on intercellular clearance and doing some derivations there, but that I think would help make the results more concrete. And I've added here an option if you saw anything else that you are interested in, we'll be happy to communicate, work on together, etc. I think that's what I had for you. If you're interested, then you can, and you have the connection, but I guess this will also be available online. So the people online, you're welcome to look at the current draft by looking at this QR code. Thank you very much.
Thank you both for wonderful talks. And let me, Reinhard, is there any business that we need to attend to before we go to questions?
If there's time, we could talk a little bit about a suitable time for extracurricular talks like the one we had this morning by Rita Colwell. There were relatively small attendance, and probably it was because it was just an unfortunate time. So maybe we could get some feedback on what good times would be, and we can do this now, or people can just contact us by email.
Yes, this was today we had an experiment where we had an extra talk at 11, and probably two meetings of the same working group in the same day was too much. But Rita Colwell gave a wonderful talk this morning, and of course these things are recorded. I don't want to take time away though from our speakers today, so let me turn it over and open the floor to questions for two fascinating talks about how much detailed quantitative measurement we can make about viruses, and also how much we don't know about viral replication. So please, questions. And again, I encourage those who don't normally ask questions to be the first in.
I can take a minute. Just there was a question in the chat about zoonotic reservoirs. So we've been playing around with that. It also somehow cosmically connects with the census of life on Earth that we're doing for the global mammals. For example, what's the overall mass of bats in the 1,000 species of bats around the world? And I think humans dominate in the current situation, where we have about 1 million infections per day across the world. I think the reservoir in humans is much larger. Saying by how much is it larger in terms of how many orders of magnitude, that's very challenging. But if anybody's interested in discussing that more, we're interested in that as well.
John, are you raising your hand?
Yeah, I did. Wait a minute, I got a feedback problem here. The zoonotic thing is our working group is trying to build a zoonotic subgroup, and I would really like to talk to you.
I think many of us are interested in the differences and similarities between the infection of a particular virus in humans and host reservoirs. And so that would be fascinating to talk about. I wanted to maybe ask a question, both of you, because both of you showed data that are related to it, which is questions about how you understand superinfection, both in terms of whether, for a single virus, whether the infection in a single cell is initiated by a single virion or multiple ones, and if a single one, what do we know about how the cell then excludes subsequent infection? What data do we have about multiplicity of infection within single cells? And then potentially, specifically, John had those beautiful, very tantalizing pictures of tracking strain competition data, if any, on superinfection by multiple viruses into single cells. So either or both of you, I think, have things to say maybe to that.
Sure, I can start off. We've done most of our experiments on a rabies-like virus called VSV, vesicular stomatitis virus. It's a single-stranded RNA virus of 11 kilobases. And on multiplicity of infection, we've done infections up to 100 virus particles per cell, where you can do the quantitative PCR and find the genomes inside the cell. So what you put on the cell, you can see inside the cell. So you do 100, you do 10, you do 1, and those sorts of treatments you can find inside of the cell. And we've done things like disable cellular translation. So there's no protein synthesis, and you can just watch the viral genome after it has entered the cell. You can see the degradation rate. It just degrades. In the presence of protein synthesis, of course, you make the polymerase and you amplify that genome. So that's an example, at least, where you can, what you put in is what you also, what you put onto the cells is also what you find inside of the cells. I should say on the studies where we've done the single virus particle on single cells at low multiplicity, then a fraction of the cells that show GFP. So this means that the virus genome gets in and it makes detectable GFP. Of those, up to 20 or 30% do not go on further to make any detectable virus particles. So you think about viruses getting in at a small one, or let's say one genome that gets in, and a sizable fraction of those that make genes that actually synthesize detectable protein, they don't go on and go all the way through the cycle to be productive. So there is probably a degradation, and some stochastic simulations suggest that, yeah, there can be degradation before you are able to amplify. This has also been found by Udo Reichel's group studying influenza, single cell infections. So that's a kind of two extremes, from one particle, one genome per cell, up to 100 genomes per cell. I'd have to think a little bit more about some of our data on the activation of innate immune responses, but we have done some on the two-color side, where we look at the viral color and we look at the host response color. I can show some further slides later on, maybe that might be of interest.
Can I jump in? What I think is a related question is, hi, this is Fred Adler. So it seems, I've gotten the impression that for some viruses, a huge fraction of produced virions are not viable. You know, either they don't have RNA or they're just not assembled properly. Can you get estimates of those? And what do they look like for coronaviruses compared to some other viruses?
Ron, do you want to take that one? I think that might have been one of your questions, actually.
Yeah, so I have it as a question, but maybe John, you have it as an answer.
I'm afraid I don't, but that's one of the burning questions for us, because I'd love to understand that 10 to the 4th. So the 10 to the fourth, you can experimentally, you can go after. I would love to see what fraction of those 10 to the fourth are actually inside particles versus free RNA. So you can treat samples with RNase, which will digest any free RNA. And generally, the packaged virus particles are protected from that RNase treatment. So if you isolate then virus particles following RNase treatment, then you can find total packaged RNA in those particles. And what you would like to do is count those particles. You can do it under the transmission electron microscopy, or you can use this kind of tunable resistive pulse sensing that we've done some work with to count. It's a kind of flow cytometry-like approach for virus particles. So that gives you particles, but then you also want to count infectious particles, and that's where you use a plaque assay. So many of you might know that standard virus textbooks like Flint in their first chapter have a table of various viruses and particle to PFU ratios that are all over the map, up to thousands or tens of thousands of particles per plaque forming units, meaning that only a small fraction of those particles that are made are actually infectious. So I don't know what the number is for SARS coronavirus. I do know that for mouse coronavirus, there have been studies in the 80s and 90s. I don't know what the numbers are on those, but they definitely found defective interfering particles on those. Yeah, I guess that's about what I can say about it. The experimental methodologies are there to figure out this, and for us, it's one of the first measures. We're starting to grow up human coronavirus now in the lab. Nan Jiang and Huichang Shi, who are in the audience, are leading that effort. And one of the things we want to do is try to make this kind of measurement.
Could I make a comment?
Absolutely.
John, I don't know if you know Ray Pickles and Rick Boucher and Ralph Barrick at UNC.
Yeah.
But they have told me with some degree of confidence that the SARS-CoV-2 will, when it infects, say, a ciliated cell in the upper respiratory tract, that it generates 50,000 copies a day with only 4% of them infectious. So those are the bounds that they give to us for the modeling that we do.
That's great. Is that copies of RNA or is that copies of particle? Are those particles?
I don't know the answer to that question. What they're saying is that 2,000 infectious virions a day for 3 to 5 days per infected cell.
That's great to know. That's definitely a group I would trust. So yeah, that's great. Have they published that?
I doubt it.
Okay. All right.
There's a paper by Barrick and Boucher in Cell last year that will have as much data as they published at that point, but I don't know about any since then.
I appreciate that. Thanks very much. I had a question actually for Ron. You focused a lot on the mutation rates, and for the coronaviruses, it has this exoribonuclease, which seems to cause extensive recombination. So I'm just wondering, my sense is, so this is from the Dennison group, which is also, I think, very well established in the coronavirus area, that they find extensive defective or genomes that are lacking many essential genes, which might be a signature of potential defective particles. I wonder if you've thought about trying to estimate sorts of maybe some of the numbers. I guess that 10 to the 4th is probably based on not full genome sequencing, but maybe PCRs on a particular gene, in which case it's conceivable that these could be defective genomes that are generated by the exoribonuclease. Have you looked into that aspect, the recombination aspect on the coronaviruses at all?
Thanks. We have not looked into that specifically. And now that you mentioned that, we will, and we could maybe afterwards and see maybe we could explore that further.
Yeah.
This is a great set of questions.
Yeah, this is Alan Carlson. Can I make a few comments?
Go ahead, Alan, please, go ahead.
Yeah, so this a couple of comments. So one on this particle to infectious particle ratio. That's been looked at, in HIV and SIV, again, by doing, PCR to count virions and then doing TCID 50 or plaque forming units. And also by diluting serum after you've done PCR to multiple folds and seeing if you have infectious units, at least in animal models. One of the things that's been noticed there is that very early in primary infection, the ratio of infectious particles to total is very high. And that with time of infection, even a few weeks or even a week or two, that ratio goes down. And it's maybe correlated with making antibody responses and generating neutralizing antibody or just having autologous antibodies, that's binding to the virions. At least in one set of experiments using SIV, if they took the plasma, and they asked whether or not if you change temperature, for example, is this property heat stable or not, which they were using temperatures at which immunoglobulins would be stable. It seemed to be heat stable. There are other plasma proteins that also, at least in HIV, people have thought may be involved in blocking infection just by binding to the surface of virions. So one of the concerns, at least in SARS, since most of the samples one gets is after people become symptomatic and possibly even after they're hospitalized, we could be looking at samples that are 2 weeks into the infection or so, at which point at least some of these effects may be relevant, in addition to all the genetic effects that go on. The other thing, Ron, in terms of about the virus being produced, at least looking at clinical samples from patients who've been hospitalized and measuring the viral loads, near the time of admission, there's a huge variation in that. One of my collaborators, Jeremy Gage, has a paper last month in PNAS where they looked at 655 hospitalized patients and have viral load data in that paper. It's NEONT, I'll send you the reference. And the data on admission, if you look at their plots, go from a few hundred copies per ml, and these are nasal pharyngeal swabs, up to almost 10 to the 12th per ml. And they do have patients up there, 10 to the 10th, 10 to the 11th, 10 to the 12th, that sort of peak viral load. If you take a median or so, it's probably more like 10 to the 8th, but there are examples of very high viral loads. And again, they may have missed the peak when they've gotten them. So that's per ml on a swab, not total lung or anything else. So I'm a little skeptical, at least of the maximum amount of virus that can be produced in a person. I'll send you that paper. And there's some others also that have very high ratios. I've also seen and done calculations on this 10 to the 4th between infectious and non-infectious, just on papers that have been published where they give RNA and they also give plaque forming units. And I've corresponded with some people who've done work in non-human primates and something like that they've measured also in their stock viruses.
Let me ask you, Alan,
Sure.
or others in the audience here who might have a better sense here. You mentioned the viral load, estimating it by the PCR, which I guess the question for me is that, I guess the distinction is here, free RNA versus particle encapsulated and protected RNA. So viral load, yeah, it's easy to do PCR, so you can quantify a number of RNA strands very nicely. But if you're quantifying those from a patient sample, then they might be free in solution or released by broken lysed cells rather than actually packaged into virus particles. And I think in general, people don't go to the go to the trouble of trying to find the particles, especially if they're non-infectious. But I wonder what fraction of actual RNAs get packaged, and then what fraction of those packaged ones.
Yeah, I don't know that answer. I mean, you mentioned that before, and I think it's be a nice experiment to do. It's not that difficult for a lab to, do the RNAs first, but I don't know of anybody who's done that.
Yeah. Okay. Good.
I know the other thing that people have been concerned with, and they're taking clinical samples, which are usually swabs, and doing PCR, that they may be measuring a virus that's actually in cells, so that when they do those swabs, they're not only getting the fluid, but they're also swabbing off cells.
Yeah.
And some groups have started also doing PCR on host genes to try and get some idea of how many cells they've isolated in that sample, and whether they're looking again at intracellular virus that's being isolated in their PCR procedures, where they do have to break open the membranes, either the viral or the cell membranes.
Yep. Okay.
So,
Good. Thanks.
Another confounding issue.
Yeah.
So, sorry, let me break in for just a second. We're at the canonical hour, so the official part of the meeting is over, but that we have a half an hour to discuss.